Good morning all. Today I am going to with the topic structure of chromosome. It include structure of chromatin, heterochromatin, euchromatin, unique and repetitive DNA, and some specialized DNAs, junk DNA, satellite DNA, and selfish DNA. First discuss with the structure of chromosome. Chromosome means chroma means color, some means body. A chromosome is a thread-like self-replicating genetic structure containing organized DNA molecule package found in the nucleus of the cell. In all type of higher organisms, the well-organized nucleus containing definite number of chromosomes of definite shape and size. The somatic chromosome number is the number of chromosomes found in somatic cell and is represented by 2n. The gametic chromosome number is half of the somatic chromosome number is represented by n haploid. Chromosomes are two types, autosomes and sex chromosomes. Autosomes that control characters other than sex characters or carry genes for somatic characters. Sex chromosomes involved in sex determination and sex characters. Two types are autosomes and sex chromosomes. Chromosome size is measured at mitotic metaphase, generally measured in length and diameter. മൈറ്റോട്ടിക് മെറ്റാഫേസിൻ്റെ സമയത്താണ് ക്രോമസോമിൻ്റെ ലെങ്ത്തും ഡയമീറ്ററും നമുക്ക് മെഷർ ചെയ്യാനായിട്ട് സാധിക്കുന്നത്. പ്ലാന്റ്സ് യൂഷ്വലി ഹാവ് ലോങ്ങർ ക്രോമസോം ദാൻ എനിമൽസ്. ദ ഷേപ്പ് ഓഫ് ക്രോമസോം ഈസ് ജനറലി ഡിറ്റർമൈൻഡ് ബൈ ദ പൊസിഷൻ ഓഫ് ദ സെൻട്രോമിയർ. സെൻട്രോമിയറിൻ്റെ പൊസിഷൻ അനുസരിച്ചിട്ട് ക്രോമസോം 3 തരത്തിൽ കാണാനായിട്ട് സാധിക്കും. ക്രോമസോം generally exist in 3 different shape: rod shape, J shape and V shape. Structurally, chromosome consists of 7 parts: centromere, chromatid, secondary constriction and satellite, telomere, chromomere, chromonema and matrix. This is the structure of chromosome. First one is centromere. Centromere, it is a localized region of the chromosome with which spindle fibers attached is known as centromere or primary constriction or kinetochore. This central part is centromere. Next, chromatid. One of the 2 distinct longitudinal subunits of a chromosome is called as chromatid. Chromatid are 2 types: sister chromatid and non-sister chromatids. Secondary constriction. Some chromosomes exhibit secondary constriction in addition to primary constriction. The chromosomal region between telomeres is called as satellite or trapent. The chromosome having satellite is called as satellite chromosome. Telomeres. The 2 ends of chromosome are called as telomeres. Telomeres are highly stable and they do not fuse or unite with the telomere of other chromosome in chromosome is constant. Chromomere. One of the small bead-shaped and heavily staining mass of coiled chromatin that are linearly along the chromosome. ഈ കാണുന്നതാണ് ക്രോമാറ്റിഡ്. ഇതിൻ്റെ എൻഡ് പോർഷനാണ് പറയുന്നത് ടീലോമിയർ. ഇതിൻ്റെ ഉള്ളിൽ ലോഞ്ചിറ്റ്യൂഡിനലായിട്ട് നമുക്ക് കാണാൻ പറ്റും, അതാണ് ക്രോമോമിയർ. ബീഡ് ഷേപ്പ് സ്ട്രക്ചർ കാണാൻ പറ്റും, അതാണ് ക്രോമോമിയർ. Chromonema. Thread-like coiled structure found in the chromosome and chromatid are known as chromonema. matrix. It is a fluid part in which chromosomata are embedded in is called matrix. ഇതിൻ്റെ ഉള്ളിലുണ്ടാവണ ആ ഭാഗത്തിനാണ് പറയുന്നത് മാട്രിക്സ് എന്ന്. These are the 7 part of the chromosome: centromere, chromatid, secondary, constriction and satellite, telomere, chromomere, chromonema and matrix. Chromosome and chromatin. An average human cell containing about 6.4 billion base pair of DNA divided among 46 chromosomes, each chromosome containing a single continuous DNA molecule. The total length of DNA of a single human cell approximately 2.2 meters long. Chromosomes are composed of DNA and associated protein together called chromatin. Structural organization of eukaryotic chromosomes. In eukaryotic cells, DNA is associated with about an equal mass of histone proteins in a highly condensed nucleoprotein complex called chromatin. In building block of chromatin is the nucleosome, consists of a histone octamer around which is wrapped about 147 base pair of DNA. The orderly packing of eukaryotic DNA depend on histones. It is the structure of chromatin. This is chromosome. This chain-like structure is chromatin. This chromatin made up of nucleosomes. This yellow portion is complex is nucleosome. This nucleosome made up of histones and this histone wrapped with DNA. This nucleosomes has histone octamer and it is wrapped with DNA helix. Chromosomes are made up of DNA and two types of proteins called histones and non-histones. These are chromatins. This chromatin fiber is made up of nucleosomes. This nucleosomes has histones octamer and it is wrapped with DNA. Both proteins are necessary for physical structure of chromosome. Histones most abundant proteins. A remarkable group of small proteins that possess an unusually high content of the basic amino acids arginine and lysine. There are 5 types of histone named H1, H2A, H2B, H3 and H4. 5 type histone ഈ ന്യൂക്ലിയോസോമിൽ ഉണ്ട്. അതാണ് H1, H2A, H2B, H3 and H4. Amount of histone is equal amount of DNA in chromatin. Histone play some vital role in eukaryotes in organizing DNA in chromosomes. Non-histone proteins are all proteins associated with DNA except histones. Example: proteins that bind to DNA during DNA replication, repair, transcription, gene regulation, recombination, etc. Each eukaryotic cell will have different non-histones. There are 3 stages in chromatin. First, nucleosomes are found by the wrapping of DNA around the histone protein. Second one, multiple histone wrap into a 30 nanometer fiber consisting of a nucleosome. Third one, higher level DNA packaging of the 30 nm fiber into the metaphase chromosome. ഇവിടെ പറയുന്നത് ക്രോമാറ്റിൻ്റെ സ്ട്രക്ചറാണ് ഇവിടെ പറയുന്നത്. ക്രോമാറ്റിൻ ഈ ചെയിൻ ലൈക്ക് സ്ട്രക്ചർ ഇറ്റ് ഈസ് മേഡപ്പ് ഓഫ് ന്യൂക്ലിയോസോംസ്. ന്യൂക്ലിയോസോംസിൽ ഹിസ്റ്റോൺ ഒക്ടാമർ അതിനെ റാപ്പ് ചെയ്തിട്ട് DNA ഹെലിക്സ് കാണാനായിട്ട് സാധിക്കും. 5 ടൈപ്പ് ഹിസ്റ്റോൺ ഉണ്ട്. H1, H2A, H2B, H3 ആൻഡ് H4. Nucleosome structure. Nucleosome model of chromosome is proposed by Roger Kornberg in 1975. Nucleosome is the lowest level of chromosome organization in eukaryotic cell. Nucleosome model is scientific model which explain the organization of DNA and associated protein in the chromosome. It is the most accepted model of chromatin organization. Nucleosome are the fundamental packing unit particle of the chromatin and give chromatin a bead on a string appearance in electron micrographs taken after treatment that unfold higher order packing. ഈ ക്രോമാറ്റിനിൽ ബീഡ് ഓൺ സ്ട്രിങ് ആ ഒരു അപ്പിയറൻസിലാണ് ന്യൂക്ലിയോസോംസ് അറേഞ്ച് ചെയ്തിട്ടുണ്ടായിരിക്കുക. ഇൻ യൂക്കാരിയോട്സ് DNA ഈസ് ടൈറ്റ്ലി ബൗണ്ട് ടു ആൻ ഈക്വൽ മാസ് ഓഫ് ഹിസ്റ്റോൺസ് വിച്ച് സെർവ് ടു ഫോം എ റിപ്പീറ്റിംഗ് എറേ ഓഫ് DNA പ്രോട്ടീൻ പാർട്ടിക്കിൾ കോൾഡ് ന്യൂക്ലിയോസോംസ്. Histones plays a crucial role in packing this very long DNA molecule in a orderly way into nucleus only a few micrometers in diameter. Each nucleosome is a disc-shaped particle with a diameter of about 11 nanometer and 5.7 nanometer in height, containing 2 copies of each 4 nucleosome histones H2A, H2B, H3 and H4. The histone octamer forms a protein core around with the double-stranded DNA helix is would 1.75 times containing 146 base pair. The DNA double helix is wrapped as superhelical left-handed then around the histone octamer core. The solenoid model represents the organization of nucleosome within the 30 nm wide chromatin fiber. The term solenoid itself defining the widening of the DNA helix. ഇതിനെ സോളിനോയിഡ് മോഡൽ എന്നും കൂടി പറയും. DNA ക്രോമാറ്റിൻ ഫൈബർ അറേഞ്ച് ചെയ്തേക്കുന്ന മോഡലിനെ സോളിനോയിഡ് മോഡൽ എന്നും കൂടി പറയും. ഇറ്റ് ഈസ് ദ ന്യൂക്ലിയോസോം. ദിസ് ഗ്രീൻ പാർട്ട് ഈസ് ഹിസ്റ്റോൺസ് octamer. This it is histone H1 histone. It is wrapped. Red portion is DNA. It is the structure of nucleosome. Chromatin is made up of nucleosomes. In chromatin, the DNA extends as a continuous thread from nucleosome to nucleosome. Each histone core is encircled by 1.7 turns of DNA, represent about 146 base pair. The H1 histone stays outside the histone octamer. The H1 histone plays a very important role in the formation of the 3 nm fiber. 2 nitrogen containing bases that pair together to form the structure of DNA. The four bases in DNA are adenine, cytosine, guanine, and thymine. These base from specific pairs, A with adenine with thymine and guanine with cytosine. It is the structure of chromosome. This is the cell. This is the chromosome. This chain-like part is chromatin fibers. It is the DNA helix part, nucleotide base. And next, histone modification. Acetylation, methylation, phosphorylation, and ubiquitination. Four types of histone modifications. First, acetylation. Acetylation means addition of acetyl group on lysine or arginine. Methylation, addition of methyl group on lysine or arginine. Phosphorylation means addition of phosphate group on serine and threonine. Ubiquitination means addition of ubiquitin group on lysine or other chains. This histone modification influence chromatin structure by regulating the binding of histone tails to other less abundant chromatin associated proteins. The reversible acetylation and deacetylation of lysine residues in the N-terminal tails of the core histones regulate chromatin condensation. Next, heterochromatin and euchromatin. Chromatin can be divided into two types of materials, heterochromatin and euchromatin. Chromatin two type ആണുള്ളത്, heterochromatin and euchromatin. First, euchromatin. Euchromatin is lightly packed form of chromatin that is rich in gene concentration. It is the euchromatin. It is a bead-like structure. It is lightly and or loosely packed chromatin. If it is often under active transcription, the structure of euchromatin's remnants of an unfolded set of beads represent nucleosomes. Nucleosome consists of 8 proteins known as histones with approximately 147 base pairs of DNA wood around them. In euchromatin, the wrapping is loose so that the raw DNA may be accessed. The basic structure of euchromatin is an elongated open 10 nm microfibril as noted by electron microscopy. Histone acetylation results in loose packing of nucleosome. Transcription factor can bind the DNA and gene are expressed. Histone acetylation loose packingന് ഹെൽപ്പ് ചെയ്യും. ഇതാണ് യൂക്രോമാറ്റിൻ്റെ സ്ട്രക്ചർ. ഫസ്റ്റ് വൺ, ബീഡ് ഓൺ എ സ്ട്രിങ് ആയിട്ടാണ് ന്യൂക്ലിയോസോംസ് അറേഞ്ച് ചെയ്തിരിക്കുന്നത്. ലൂസിലി അറേഞ്ച്മെൻ്റ് ആണ് യൂക്രോമാറ്റിൻ. നെക്സ്റ്റ്, ഹെറ്ററോക്രോമാറ്റിൻ. The region of chromosome that appear relatively condensed and stained deeply with DNA specific strains. It is tightly packed form of DNA, transcriptionally inactive. Euchromatin transcriptionally active ആണ്, heterochromatin inactive ആണ്. Methylation of DNA and histone cause nucleosome to pack tightly together. Transcription factor cannot bind the DNA and genes are not expressed. This is heterochromatin. It is tightly packed nucleosomes. In euchromatin, histone acetylation result in loose packing. In heterochromatin, methylation is result in tightly packing. Heterochromatin are two types: constitutive heterochromatin and facultative heterochromatin. Constitutive heterochromatin. Constitutive heterochromatin is the stable form of heterochromatin. It does not loosen up to form euchromatin. It contains repeat sequence of DNA called satellite DNA. It can be found in centromere and telomere and throughout the chromosome in major amount as well. It is made of satellite DNA and is usually involved in structural function. Second one, facultative heterochromatin. Facultative heterochromatin is reversible. Its structure can change depending on the cell cycle. It may convert to euchromatin. It is characterized by the presence of another kind of repeated DNA sequence known as line sequence. Example is the inactivated X chromosomes of females. Two types of heterochromatin: constitutive heterochromatin and facultative heterochromatin. Properties of heterochromatin. Heterochromatin is condensed. Heterochromatin DNA is late replicating. Heterochromatin DNA is methylized. Heterochromatin does not participate in genetic recombination. Heterochromatin is transcriptionally inactive. This is the chromosome. This condensed and tightly packed nucleosome part is heterochromatin. Loosely packed nucleosome, this part is euchromatin. Next, chemical composition of chromosome. Chemically, chromosomes are nucleoprotein in nature, means are composed of DNA, RNA and protein. Generally, chromosome contains 30 to 40% DNA, 50 to 65% protein and 0.5 to 10% RNA. First, DNA. The amount of DNA is present in somatic cell is constant. The DNA content of gametic cell is half of the somatic cell. DNA chromosome is two types: unique DNA and repetitive DNA. Unique DNA. Unique DNA consists of those DNA sequences which are present in a single copy per genome and are unique in nature. Unique DNA, also known as non-repetitive DNA, code for protein which require in large quantity for cell, example storage protein. Next, repetitive DNA. Repetitive DNA consists of DNA nucleotide or base sequence which are few to several hundred base pair long and are present to several to a million copies per genome. Human genome contains 30% repetitive DNA. Repetitive DNA further divided into highly repetitive DNA and moderately repetitive DNA. Next, RNA. Chromosome, nucleoprotein protein in nature means composed of DNA, RNA and protein. Discuss DNA. The next RNA. RNA purified chromatin contain 10 to 50% RNA. RNA associated with chromosome in messenger RNA, transfer RNA and ribosomal RNA. Protein associated with the chromosome is classified into two broad groups. First, histone or basic protein. Histone constitutes about 80% of total chromosomal protein. They are present in an almost 1:1 with DNA. Non-histone protein make up to 20% of the total protein mass. Content of non-histone protein is different from species to species. Next, junk DNA. Next topic is junk DNA. In genetics, the term junk DNA refers to region of DNA that are non-coding. DNA-യിൽ non-coding ആയിട്ടുള്ള ഭാഗത്തിനാണ് ജങ്ക് DNA എന്ന് പറയുന്നത്. Non-coding regions. DNA contains instructions that are used to create protein in the cell. The amount of DNA contained inside each cell is vast, and not all of the genetic sequences present within a DNA molecule actually code for a protein. Only about 1% of DNA is made up of protein coding genes, and other 99% is non-coding. 99% non-coding ആയിരിക്കും. Non-coding regions ആയിരിക്കും. 1% മാത്രമായിരിക്കും കോഡിംഗ് റീജിയൻസ് ഉണ്ടായിരിക്കുക. ഈ non-coding regionsനെയാണ് പറയുന്നത് ജങ്ക് DNA. Non-coding DNA does not provide instructions for making protein. Scientists once thought non-coding DNA was junk with no known purpose. ജങ്ക് DNAക്ക് ഒരു സ്പെസിഫിക് പർപ്പസ് ഉണ്ടായിരിക്കില്ല. Some of this non-coding DNA is used to produce non-coding RNA components such as transfer RNA, regulatory RNA and ribosomal RNA. In human genome and the mice genome diverged from a common ancestor around 65 to 75 million years ago. However, the protein coding DNA only makes about 20% of conserved DNA. This genome with the remainder of the conserved DNA found in non-coding regions. This is the junk DNA. This non-coding part is junk DNA. The portion of DNA which is known as junk DNA come into existence through transposition. Transposition is the movement of section of DNA to different positions in the DNA. If this transposition activity happens in the cell, that makes egg or sperm the resultant. This simply means with the passage of time and successive transpositions in offsprings, the amount of junk DNA will continue to increase. This is the reason why earlier species has less junk DNA compared with the later species. These are non-coding DNAs. Satellite. Some Non-coding DNAs are satellite DNA, telomeres, introns. Non-coding RNA genes, gene regulatory sequences. Next, satellite DNA. Satellite DNA consists of very large array of randomly repeating non-coding DNA. Non-coding DNA തന്നെയായിരിക്കും സാറ്റലൈറ്റ് DNAയും, അത് റാൻഡംലി റിപ്പീറ്റഡ് ആയിരിക്കും. Satellite DNA is the main component of functional centromeres and form of main structural constituent of heterochromatin. The name satellite DNA refers to the phenomenon that repetitions of a short DNA sequence tends to produce a different frequency of the bases adenine, cytosine, guanine and thymine, and thus have a different density. Classification of satellite DNA. It is classified into two categories on base composition, length of segment and number of repetitive unit. റിപ്പീറ്റേറ്റീവ് യൂണിറ്റിൻ്റെ നമ്പർ അനുസരിച്ച്, അതുപോലെ സെഗ്മെൻറ്റിൻ്റെ ലെങ്ത് അനുസരിച്ചിട്ട് സാറ്റലൈറ്റ് DNA ക്ലാസിഫൈ ചെയ്തിട്ടുണ്ട്. ഫസ്റ്റ് മൈക്രോ സാറ്റലൈറ്റ് ആൻഡ് മിനി സാറ്റലൈറ്റ്. മൈക്രോ സാറ്റലൈറ്റിനെ നമ്മൾക്ക് സിമ്പിൾ സീക്വൻസ് റിപ്പീറ്റ്സ് അല്ലെങ്കിൽ ഷോർട്ട് ടാൻഡം റിപ്പീറ്റ്സ് എന്നും കൂടി പറയാം. മിനി സാറ്റലൈറ്റിനെ VNTR, വേരിയബിൾ നമ്പർ ടാൻഡം റിപ്പീറ്റ്സ് അതും കൂടി പറയാം. ഫസ്റ്റ് മൈക്രോ സാറ്റലൈറ്റ് composed of repeated unit of 2 to 10 nucleotide pairs. The repeated sequence is often simple, consisting of 2, 3 or 4 nucleotides. One common example of a microsatellite is CA and repeats where n varies. ഇതിൽ CA, സൈറ്റോസിൻ ആൻഡ് അഡിനിൻ ഇത് റിപ്പീറ്റ് ചെയ്തിട്ട് വരുന്നതാണ്. അങ്ങനെ സിമ്പിളായിട്ടുള്ള സീക്വൻസിനെയാണ് മൈക്രോ സാറ്റലൈറ്റ് എന്ന് പറയുന്നത്. അടുത്തത് മിനി സാറ്റലൈറ്റ് VNTR, വേരിയബിൾ നമ്പർ ടാൻഡം റിപ്പീറ്റ്സ്, composed of repeated unit of 10 to 80 nucleotide pairs. മൈക്രോസാറ്റലൈറ്റ് 2 to 10 ന്യൂക്ലിയോട്ടൈഡ് പെയേഴ്സ് റിപ്പീറ്റ് ചെയ്തിട്ട് പോകും. ഇതാണെങ്കിൽ 10 to 80 ന്യൂക്ലിയോട്ടൈഡ് പെയേഴ്സ് റിപ്പീറ്റ് ചെയ്തിട്ട് പോകും. These occur at more than 1000 locations in the human genome. Consist of repetitive, generally GC-rich variant repeats that range in length from 10 to over 80 base pair. Some contain a central sequence of GGG, CA, GGA, NG. ഈ സീക്വൻസ് റിപ്പീറ്റ് ചെയ്തിട്ട് ഉണ്ടാവും. മിനി സാറ്റലൈറ്റിൽ, മൈക്രോ സാറ്റലൈറ്റിലാണെങ്കിൽ CA ഇതായിരിക്കും റിപ്പീറ്റ് ചെയ്തിട്ട് ഉണ്ടാവുക. യൂസസ് ഓഫ് VNTR ആൻഡ് STR. ഹൈ ഡിഗ്രി ഓഫ് പോളിമോർഫിസം. They are used as molecular markers for relationship studies, extensively used in DNA profiling. Next, selfish DNA. Transposable elements are often termed selfish DNA because they are parasitic DNA sequences that inhibit a host genome. Over time, many copies of selfish DNA are inactivated by mutations and deletions, leaving DNA remnant called junk DNA. Selfish genetic elements are genetic segments that can enhance their own transmission at the expense of other gene in the genome, even if this has no positive or a net negative effect on organismal fitness. It is the selfish DNA. This is the chromosome. These are the selfish DNAs. DNA that merely inhibit the genome of other organisms has been named selfish DNA because it behaves as if motivated by its own interest, not those of the host DNA. Selfish because their only function seem to make more copies of themselves, and junk because there is no obvious benefit to their host. There are the three DNAs: selfish DNA, satellite DNA, and junk DNA. And these are the structure of chromosomes. Thank you.